Hello everyone and welcome to the biology unit of Year 10 Core Science. Before we begin, I would like to take this opportunity to discuss this, which I'm sure you know by now is a representation of the coronavirus, and it is the reason why we were staying at home and why I'm talking to a computer screen recording my lecture. And in the past few weeks, I've noticed a lot of science and specifically biology related terms popping up on headlines and YouTube videos. I mean, I say headlines, but I don't know whether anyone read the newspaper anymore. So I'm just going to give you a few examples of these headlines. So COVID-19 patients need to be tested for bacteria and fungus and not just virus. So we have bacteria and fungi and virus. So what is the difference between the three types of organisms? Are they organisms? What are they? What are bacteria? What are fungi? What is the difference between bacteria and fungi compared to a virus? And then you have a lot of talks about things like antibiotic resistance. So what is an antibiotic? What does it actually do? And then obviously you have seen this hydroxychloroquine or chloroquine. Now this is a anti-malarial drug, so it's a drug to treat malaria. And then you're like, oh, so what is malaria? What is the difference between malaria and bacteria and fungi and virus? And then we'll talk about vaccine. A lot of talk about vaccine. So what does a vaccine actually do? I do hope you know by now. And then you will see a lot of information and headlines and videos on coronavirus test. We need more tests, we need better tests, we need faster tests, we need to make sure that everyone have access to these tests. But what I want to really be able to answer is what exactly goes into the coronavirus test. So when we say you're going to get tested for coronavirus, what exactly are you being tested for? And then we have, so eventually later on there is antibody test, which is different from the original coronavirus test. So what is the difference between the first test and the antibody test, and why is it not guaranteed that you have coronavirus, even if the test comes out negative that you don't have coronavirus? And then we'll talk about past pandemics. So things like the Black Plague came up, and then the influenza, Spanish flu at the outbreak of World War II, and then occasionally Ebola came up as well. So there are a lot of terms here, and I really think that this is the perfect opportunity to use this unit on biology to clear up some of the terminologies that you may see in the news, and hopefully it allows you to understand the situation that we're in right now a little bit better, and also to appreciate the role of biology in real life. I don't think there is a better time than now for you to understand why you need to get biology. So you could actually figure out all of these headlines and don't get bombarded and terrified by what is being given to you. So we're going to use the next few weeks to maybe provide some clarification for some of these headlines. I'm not going to be able to talk about everything just because of the scope of the subject and also the limitations of my knowledge. But I really do hope that you understand a little bit more about these headlines and coronavirus and viruses and pandemics than you are right now. So this lesson today, this is a question that I want to answer. What is the difference between the coronavirus and bacteria and fungi and malaria and human? So that is the question that I want to answer for these two periods. And in order to answer this question, I would need to cover these dot points. So this is the actual knowledge that we're going to cover to answer this question, is to describe the biological levels of organisms. And then we're going to talk about organelles. We're going to talk about functions of the organelles and distinguishing between prokaryotes and eukaryotes and plant and animal cells based on the structure of the cell. A lot of this, if not all of this, you should have learned in year 8, so this should be a very easy breezy lesson that is a recap of everything you've learned before, but we'll see about that, I guess. All right, so we're going to start at where we end with chemistry. So the whole focus of chemistry, well, not the whole focus, but the main focus of chemistry is on the atoms. And one definition of atoms is the smallest building block of matter. So you can have protons, you can have neutrons, actually neutron. Let's say this is a neutron inside a nucleus, and then you can have the electrons on a shell surrounding the nucleus. So this is a, I guess, schematic representation of an atom. And then you can have things like molecules, and that's when you have multiple atoms combined together. So you can have, for example, H2O. This is an example of a molecule which consists of one oxygen atom and two hydrogen atoms. Or you can have CO2 gas. So the study of atoms and molecules and the connections and bonding between atoms and between molecules, that's a really big part of chemistry. And I think that's the part of chemistry that we looked at in year 10. And this is where you start to have a divider. And I probably should move this down here. So you have things like H2O, CO2, and this is an atom. This is once you have a lot of molecules, that's when you transition from chemistry to biology. So macromolecules or biomolecules is another name that you might have seen. Essentially, they are really, really large molecules and they consist of many, many atoms. But other than that, there's virtually no difference between a molecule and a macromolecules. Macro just mean big. So they just have a lot more atoms than, say, H2O or CO2. Examples of macromolecules could be DNA, that is a macromolecule. It contains many, many atoms. And then you have proteins, which I'm sure you have heard of in your everyday life. You also have lipid. Now, lipid is just a fancy name for fat, but then oil is also a type of lipid. And the last type of macromolecules or biomolecules would be carbohydrates or carb. So we're talking sugar and bread and potatoes, and we eat a lot of chips these days on pasta, so there's that. I feel like this is the time when we all want to seek comfort in carbohydrates. So, four types of macromolecules: DNA, proteins, lipids, and carbohydrates. And they just a lot of atoms combined. Now, if you have multiple macromolecules, they will make up what is called an organelle. And once you get to organelles, this is when you start to classify substances based on their functions. So organelles are specialized structures, and by specialized, I mean they have a specific function. So have a specific function. So an organelle is essentially a structure that has a specific function within a cell, and an organelle is going to consist of multiple macromolecules or even molecules working together. So you have macromolecules and other molecules as well. An organelle could have some fat with some protein, with some carbohydrates, maybe some DNA in it. So the classification of an organelle, it's really just because all of these macromolecules and molecules form one unit, and that unit has a specific function. So we're going to talk about different organelles in a little bit. So it's basically a combination of macromolecules and molecules. And then once you have multiple organelles together, you have a cell, and this is the smallest unit of living thing. So notice how we start the chemistry unit with an atom, which is the smallest building block of matter. Now we're going to start the biology unit with a cell, which is the smallest unit of living thing. Another way to think about it is if you go from an atom to a cell, you just have an increase in the amount of atoms that you have. So we have a lot of atoms in a cell, but fundamentally every matter should be made of atoms still. So things that you learned in chemistry is still valid. It's about the level of size and amount of atoms that you choose to deal with. That's the main difference between chemistry and biology. So increase in amount of atoms. I want to extend this classification further, and I'm going to use cell as a starting point. But before that, let's just talk about the macromolecules or the biomolecules building blocks. So there are four types: nucleic acid. Now this is your DNA and your RNA. I don't know whether you have heard of RNA, but we'll talk about RNA in a future lesson. So nucleic acid, DNA and RNA, they are the genetic materials. If you have, I'm sure you have heard of it. And the main function of DNA and RNA is to provide instructions to make proteins. So even though you may feel like DNA is really important because we've heard of it and we talk about mutation and DNA denaturing and how you can have X-Men and stuff like that, but really the protagonist of a cell is not DNA, is a protein. Protein is actually the molecule that carry out all of the functions in a cell, from fixing a cell, or allows you to move, or allow you to test things like that, or even talking, carrying a thought. Anyway, so proteins are the main player here. They are the molecules that carry out all of the functions that allow you to live. And DNA provides instructions on how to make the proteins. So that's the relationship between DNA, RNA, or the nucleic acid, as they both called, and proteins. You also have two other types of biomolecules: carbohydrates. Carbohydrates just provide energy, and lipids, which store energy. So if you think about it, it's actually pretty easy. You have the instructions, which is a nucleic acid. The instructions is to make the proteins, and then you have the energy to that can be supplied for the process of making proteins, and then you have any excess energy, you store it. So they are your four main types of macromolecules. You have to eat proteins to make proteins, and you have to eat carbohydrates. Any excess carbohydrates can be turned into lipid, and you don't need DNA. You get that from your parents. We'll talk about these macromolecules more in future lessons, but this is just a quick definition of what they are and what they do. And this is what they look like, which again, we'll talk about more in future lessons. All right, let's talk about the biological levels of organisms. So cell is the first one, which is the smallest unit of living thing. I think I'm just going to wait for this to go back. This animation is a little bit too fast. So cell is the first one, and then if you have multiple cells, you have a tissue. Multiple tissues an organ, multiple organs an organ system, and then you have an organism. And then it keeps going to population, community, ecosystem, biome, and biosphere. So a tissue is basically a group of cells that performs a specific function. So for example, muscle is the tissue. What other tissues do we have? Muscle is the only one I can think of right now. And then if you have multiple tissues with the same function, you get an organ like the heart, which is essentially just a lot of muscles. Skin is also considered an organ and is, I think, the largest organ. And it performs the same function, which is to provide a barrier between everything inside of you and the outer environment. Very, very crucial. Multiple organs will form what is called an organ system, which is a group of organs that work together to form one or more functions. So an example could be the digestive system, or the circulatory system, or the nervous system. A digestive system, I'm sure you know, is responsible for digesting food and distribute nutrients throughout the body. The circulatory system is circulation of blood. Essentially, is to circulate oxygen and then to collect carbon dioxide. And then when you have multiple organ system combined, you have an organism, which is a living entity. Now, the definition of to living is that you can grow, you can metabolize, you can respond to change, and reproduce. So if you cannot do these things, you're not considered a living organism or an organism. And then there are other higher levels of classification, which we will not talk about in details in this course, but I think we're just gonna stop at organism. We'll look at the relationships between different organisms later when we look at evolution. But for now, let's just go from cell to organism. Okay, I just want to give you an analogy to explain the difference between a virus and a living organism, because I think the first thing that you want to keep in mind is a virus is not considered a living organism. So why is that? So let's say, for example, this is my DNA, and I just mentioned before that DNA or RNA, in the case of virus, it can be in the form of RNA. They're basically instructions on how to make proteins. So if we just use this analogy and assume that the actual origami is protein, So in a virus, the virus has the instruction on how to make proteins, but that's it. It just has an instruction, and maybe that instruction is going to be protected in maybe a plastic bag. In the case of a virus, is what's called a protein code. So a virus is just a protein code, and within it you have a piece of instruction. This is a virus. And this is a problem because if you just have the instruction to make, say, origami, but if you don't have any paper, you don't have any people making the origami, then you can never make the origami at all. You just have the instruction. So the problem with a virus is that even though it has a DNA and RNA, it doesn't have any mean to make its own proteins. It can give instructions, but it can't make it itself. If you compare a virus to an actual cell, a cell will also have the instructions on how to make the origami, but it can also do it. So it has an organelle that can make the proteins. Let me just right here, it can make the protein. So you can think of it as a worker, a person. It also has the materials to make the protein. The material you can get from the environment, depending on the cell, but not only does it have the instruction, it also has the person or the hands in this analogy to make the protein. And this particular person is called a ribosome. So a ribosome is an organelle in a cell that makes the protein from the instructions. And both of these are also enclosed in some sort of barriers to separate the inside from the outside, and this is called the cell membrane. So on a fundamental level, the only difference between a virus and a cell is just virus has instruction but no ability to act on that instructions. The cell has the instruction and can translate the instruction into an actual product. And the instruction in this case is to make protein, always. So let's just put some notes into that. So a virus consists of a piece of nucleic acid, DNA and RNA. So normally when I draw a virus, I just go this like this, and it is contained in a protein code. Your scientific diagram is going to be correct if you label it, otherwise this looks like a face. So you have a protein code just to protect the DNA and RNA, and then you have the DNA or RNA, and that's it. It doesn't have anything else. So it's not considered a living thing or a living organism because it cannot reproduce, it cannot make its own proteins. It can only, it only has the instructions. So the interesting thing about a virus in general, and coronavirus will also fall under this, is that the way that a virus infects a host is all that it does is it injects the DNA or RNA. So it gives the instruction, it gives its own instruction to the host cell and manipulate the cell so that instead of producing its own proteins, the cell is going to produce the virus's proteins. So the virus just try to make use of all the facilities that the cell has to make its own product. It doesn't want to build its own building or build its own factory, I guess. It just want to go and hijack a system that's already in place, which, if you think about it from a virus perspective, seems to take a lot less work than having to manage your own system. But it just gives the instruction to a cell and tell that cell, no, you're going to make my protein instead. I mean, there are other things as well, but I'm not going to go into it in this video. I'm going to leave a link to another video if you're really interested in how a virus works, and you can watch that later. And viruses will vary in the DNA and RNA that they carry, the shape of the protein code, and the type of organisms that they infect. So these are some examples of different types of viruses. So the shape of the protein on the outside could be very different. So this is called complex shape. An example of this could be HIV. I think spherical is coronavirus or influenza. Bacteriophage is another type of virus that has a complex shape, which I've actually used before. Bacteriophage is a virus that infects bacteria. And then you have other types of virus that could be helical or polyhedral. So that's a virus, very, very simple in structure, not even considered a living thing. And then we can also formalize the definition of a cell. So unlike a virus, the main difference here is that a cell can produce its own protein. So not only does it have the instructions, it also has the means to carry out these instructions to make a product. There are three most fundamental structures. You need a membrane, You need the instruction, which in the case of a cell will always be DNA, and you need the factory to actually produce this protein, and that's an organelle called ribosome. So it can make protein from DNA. This is a very detailed diagram of what a ribosome looks like. So if you actually draw it properly, it contains like a two units. It's like a big one and then a small one, and then the genetic information come in here, and then the proteins will come out somewhere. So that's a ribosome. It really depends, but ribosomes are really, really small. So sometimes when you look at some diagrams, they just draw it like a circle. So again, this is why you need to label your diagrams. Otherwise, it's very hard for me to know. But if you need to draw the ribosome in full details, actually kind of looks like this, and like two folds, and then the RNA comes in and the protein comes out. So I can draw my cell with a membrane. So this is a cell membrane, and it's gonna have some DNA. Most of the time, well, it could be circular, it could not be. So this is a DNA, and because I don't want to draw that, I'm just gonna draw a circle because this is really, really small, and I'm just gonna say this is the ribosome. So one extra organelle makes all the difference. The cell is now sustainable in the sense that it can produce what it needs inside itself. It doesn't need to go and ask for someone else's help. I do also want to spend some time discussing membrane because I'm going to use the word membrane a lot in this video, and I think even in future videos. A biological membrane, I think a very simple way to understand what a membrane is, it's a barrier. It's a barrier that surrounds the cell, or the virus doesn't have a membrane, but it surrounds the cell or the organelles, and it is selective, so it allows some things to pass through while preventing other things. So think of it as a door that has a key pass. So you need to have the pass in order to go through, otherwise you cannot. So it's very selective, and this is just to protect the cell and the organelles from foreign invaders, and anything that doesn't have a passport or a key pass will not be able to pass through. Most biological membranes are made from lipid. This is an example of a membrane. This is a lipid part, by the way. Again, I'm not gonna go into the chemistry of it. It's a little bit complicated and a little bit beyond the scope of year 10. But I think any of you who's doing year 11 biology probably heard this before. In most cases, a double membrane is used. So this is actually a double membrane. So we tend to just draw it like one line for one membrane and another for another membrane. But sometimes you draw both and you just say this is a double membrane. So if we go back to my previous diagram, the cell membrane is actually a double membrane. So I'm just going to label it as a double membrane. I'm not going to go into why it's a double membrane. You don't need to know it really for year 10 science, but if you're really interested, you can ask me and then we'll have a conversation about that. All right, so that's membrane. So let's say that's it. And the question here is there any room for growth, or is it all there is? You just have DNA and you have ribosomes. So very simple. But if you think about different types of cells, I think one simple analogy, if we continue to use the analogy of the origami, is just if you have a lot of instructions and if you want to make a lot of origami, you really want to build some organization and systems in place to manage everything and make sure all processes are efficient. So cells can have more than one piece of DNA. Actually, what tends to happen is when the DNA gets really long, so when you have a lot of instructions like this, and I read somewhere that if you unwind, so the DNA inside a human cell is really tightly packed. It's like think of it as string, and then you coil it into a ball of yarn. But if you unwind all of that, it's around 3 meter long per cell, and you have a lot of cells in your body. Every time I think of that fact, it just blows my mind. We're going to talk about that more in, I think, the next lessons. But anyway, let's say you have a lot of instructions. So each of these is a DNA. So your DNA is now not just one piece of instruction, it's a lot. And they also more complicated than before. Anyone who's into origami making, this is what is called a crease pattern, which is like the fancy way to provide origami instructions. So when you have a lot of DNA, a lot of instructions, what you want to do is instead of having the instructions floating around in the cell, um, you probably want to put it in a safe. So you're not gonna, because if you lose the instruction, it's going to be bad. So all the instructions are placed in an organelle, and this is called the nucleus. So instead of just having the instructions flying around, you just have it in a safe or building, if you will, and it never leaves the building. If you want to provide instructions on how to make these origami, you're going to make photocopies of the instructions rather than using the original blueprint, just in case something happens to it. So you have multiple workers now because you have a lot of DNA, and let's say the demand is more and you want to make more. So you're just going to make copies of the DNA and send it to the workers, which is, if you remember, these are the ribosome. So each of these is a ribosome. So the instructions never leave the nucleus. Copies of the instructions will leave the nucleus to be sent to the ribosome. But because you now have a lot of ribosome, it's probably a good idea to, instead of having the ribosome floating around in the cell, it's a good idea to have them embedded somewhere so that it's easy to control, and you can also assist the ribosome in quality control of the proteins that they make. So what if the proteins that come out is wrong? So what we do is we're going to adhere the ribosome to another organelles. So all the ribosomes, instead of floating around, they're going to stick onto this row, if you will. You think of it as you forcing the office workers to sit in specific cubicles. So this is like a membrane that the ribosome are stick on, and this membrane is another organelle, and it's called endoplasmic reticulum. So this is the endoplasmic reticulum. And the purpose of the endoplasmic reticulum is just to control all the ribosomes and assist the ribosomes in making the products, which is protein. Now, if you have a lot of workers and you have the nucleus and you have endoplasmic reticulum, you also want to make sure that you provide enough energy to the ribosome so they can continue making proteins all the time. So it's probably better not to just absorb energy or food from the outside. It's probably better to have like a designated area where all the energy can come from. And I'm just gonna say you have a fridge or you have like a canteen inside your factory, and this is called the mitochondria, and this is where the energy supply is. So you have your worker, you supply energy to the workers, and let's say, for example, this product, which is a very, very nice, this is an origami, by the way. If this product is faulty in some way, you want to have some means to get rid of any faulty product, anything that doesn't work. Let me just rewrite the endoplasmic reticulum on this side. Endoplasmic reticulum. Such a nice name. It's abbreviated to ER. It's not emergency room though. So let's go back to this. Let's say this product is faulty. There's something wrong with it. So I need to get rid of it. So I need to have like a trash can or a paper shredder is probably better to degrade any faulty products, and I can reuse it. Actually, in the cell you can reuse the paper and to make new products. So you need to have like a trash can or paper shredder, and that is called the lysosome. Now, if you're very, very good, let's say this thing, so you produce is also an origami, and you actually want to send this product out of the cell into another cell for another cell to use it. If you want to do that, you need to have some version of I don't know, post office, so it can package the product and put a label on it and then send it away. So you need an extra organelle that is going to be responsible for packaging of your product for export. And that organelle is called the Golgi apparatus or Golgi complex. Okay, are we missing something? Yes, or sometimes some of the products just end up being used being used within the cell. In that case, it's not going to go through the Golgi. And all of this has to still be That's not good. All of this has to still be encompassed within the cell, so it's going to be surrounded by a cell membrane as well. So the reason why the structure of the cell gets more complicated than just DNA and ribosome is because you have a lot more DNA and you have a lot more ribosomes, you want to make a lot more proteins, and you want to be able to export your protein. So think of it as a business that is growing and expanding. You just need to have more structures and more specialized departments within that business. So I'm going to go through these organelles in a little bit more details now. We're going to talk about the nucleus. So nucleus, the purpose is to store the DNA of the cell, and it is a membrane-bound organelle. So there are membranes, there is one layer of membrane surrounding the nucleus, and the DNA cannot leave the nucleus. Whenever you want to obtain that instruction, you're going to come into the nucleus, make a copy of the DNA, and then take that copy out. But the DNA will always stay inside the nucleus. It's very, very important. Because if your DNA is mutated or is wrong in some ways, that is going to cause a lot of damage. It could lead to cancer and other things. So we'll talk about DNA more in the next lesson. Anyway, so this is a very cute animation of the nucleus, and it's holding your genetic material, your DNA, and I'm not entirely sure what this is supposed to represent. Maybe that's the copy, but it's the DNA doesn't leave the nucleus, is what I'm trying to say. So we can redraw our cell now. So instead of having the DNA anyway, I'm just going to draw this, and let's say that's the nucleus. Now, in a lot of diagram, what you're going to see is a nucleus, and then there's like a small thing inside called the nucleolus. And that's actually where the photocopying is made, but I don't think you need to go into that much details here. So this is just the nucleus, and you still have the ribosome. Yeah, so ribosome, and then we have the membrane. Ooh, cell membrane. So that's the nucleus. The other organelles that I want to talk about is the ER, or endoplasmic reticulum. The endoplasmic reticulum is an organelle that, even though it's an organelle, it's really just a bunch of membranes folded together, and you're going to have ribosomes embedded on it, which is why it's generally called rough ER. It's rough because of the texture. It's rough since there are ribosomes embedded on it. There is also the smooth ER, but I'm not going to talk about it. So the function of the ER, the endoplasmic reticulum, is to assist with protein synthesis and transport within the cell. So this is a rough ER. The ER, actually, the way that we draw it, we normally just have like a few folds. So that's just the ER, just to show the membrane, and you tend to always draw little dots to represent ribosomes on the surface of the ER. This one also kind of look like chloroplast, so that's not good. So this is your ribosome, and the whole thing is the ER, or endoplasmic reticulum. So you can see here, the ribosomes are embedded within the ER. The Golgi, it's very similar to the ER actually. They also made of folded membranes, but it's actually responsible for packaging protein for export. So the ER is mainly just to make the proteins and maybe any internal transport. If the protein that is going to be made would then be exported and leave the cell, then the protein has to be transferred from the ER to the Golgi, and then the Golgi is going to package the protein, put an address on the package so you know where the protein is supposed to go, and then it allows the protein to be exported. So this is the Golgi. It looks very, very similar to the ER. You probably can even draw it using the same structure. The only difference between the Golgi and the ER, well, first of all, it doesn't have any ribosomes on it, and second of all, you're going to label it Golgi, so I know it's not the ER. But they're very, very similar in structure. So Golgi apparatus, or Golgi complex, or Golgi body. There's so many names for this. Anyway, so that's the endoplasmic reticulum and the Golgi apparatus. The lysosome is your trash can or paper shredder, and it is basically just a sack. It's like a bag of digestive enzymes, very little digestive enzymes, and it's just going to eat up any fragments that are supposed to be mistakes or waste and degrade them. So this is a lysosome. I'm just going to go eat this up and then degrade it. So, oh, this poor fragment is dead now. Okay. Lysosome doesn't have any really interesting thing to draw. Normally it's just drawn as a circle and then you call it the lysosome because it doesn't have any characteristic features in its structure. It's just a bag of lethal digestive enzyme. So this is the lysosome. The last and definitely not least organelle is the mitochondria. Mitochondria is the plural form. The singular is actually mitochondrion. So I don't care which form you use. People still use mitochondria a lot, even though it's to talk about one mitochondria. Okay, so mitochondrion is singular, and the plural form is mitochondria. The mitochondrion or the mitochondria is essentially the organelle that produces energy in the form of ATP for the cell. So, well, I actually said here, it is also a membrane-bound organelle, but it's not just like a stack of membrane. It has inner and outer layer, and this looks like a Pokemon, but it produces energy is what you need to know. And there is a very distinctive drawing when you look at any drawing the mitochondria is at this bit, and always reminds me of like a baguette, and then you have, I don't know, ketchup and whatever spread on it, but you tend to have an outer layer and then you have an inner layer. So this is how I normally draw the mitochondria. As long as you label, we know what you're trying to do. So mitochondria. If you're interested in doing biology in VCE, you're going to actually look into why it has an outer layer and an inner layer and how exactly does it produce energy in the form of ATP, which is very, very interesting stuff. Mitochondria. Okay. So let's redraw our cell now. So this is the upgraded factory, the expanded version. The original version contained just some DNA with a ribosome. So ribosome, some DNA, and a membrane. So that's the three main features. You have the instructions, you can make the proteins, and everything is contained. If you have a lot of DNA, you're going to put it, so you have a lot of DNA, you're going to put it in a specialized organelle and you call it the nucleus. So this is the nucleus. You also, if you have a lot of DNA, there is a high chance that you also have a lot of ribosomes because you want to make a lot of proteins. So if you have a lot of ribosomes, you want to be able to quality control your proteins. So you're going to have all of those ribosomes, if not most of them, stuck onto this extra structure. So all the ribosomes are now here. And this structure, it's going to manage all the ribosomes and assist with protein production, and that structure is called the endoplasmic reticulum. If you make a mistake, the mistake will be, well, the mistake, the faulty product is going to be destroyed and recycled using the lysosome. So this is your paper shredder, if you will. And if you want to export your protein, you need to send it to, if you think of it as the post office, and the post office is the Golgi complex or Golgi apparatus or Golgi body. So Golgi apparatus. And all of this requires energy, and the energy is going to be in the form of mitochondria, which has an outer and an inner fold. And the mitochondria is the organelle that provides energy for your cell. And everything, all of this has to be inside. Okay, I need to, let me try to do this. All of this has to still be inside a cell membrane. So I'm just going to draw a cell membrane. So there's still a cell membrane. There are so many other structures, by the way. For example, we don't talk about the microtubule, which allows everything to stay in place and not float everywhere, but we're not going to talk about that in year 10. So this is another diagram. This is an animal cell, and you can see, obviously, my artistic talent is limited. And there are a lot of features I don't show. For example, the nucleolus. You can draw the nucleolus in if you want, but I don't think we'll talk about the nucleolus a lot. But most of the time when you see a nucleus, it has like a little bit inside the nucleolus. Okay, and then you can see the mitochondria and the Golgi apparatus, and this is the endoplasmic reticulum. The dots are ribosomes. There are some ribosomes that are free as well, that do not stick onto the endoplasmic reticulum. You have your lysosome, but then there are so many other structures. But these are the ones that I want you to be able to recognize when you look at a diagram of a cell. Okay, I'm going to put, you can print this out and stick into your notes if you want, but I don't expect you to be able to label everything like the microtubule and the centrosome and the microfilament. I'm not going to expect you to be able to do that, especially things like secretory vesicle. However, I do want to talk about cytosol and cytoplasm. Essentially, the cytosol is the liquid found inside the cell. So all of this is not dry. There's a lot of water inside the cell. Everything's sort of floating, really. So the actual liquid is called the cytosol. And the liquid with all the components is called the cytoplasm. Another definition of cytoplasm is all the components except for the nucleus. So this is a cytoplasm. I don't even know what is going on here. But anyway, so cytosol is just the liquid plus the components minus the nucleus is your cytoplasm. Okay, so we can also compare the two types of cells on the two types of factories. A prokaryote cell is this one. So you just have the instruction and you have the ribosome to make it, and that's pretty much it. Whereas a eukaryote cell, you have a lot more instructions. So the instructions are inside what is called a nucleus. This is not the case in the prokaryotes. And then you have so many other organelles which I can't fit into this slide, so I didn't show them here. But they all encompass the cell membrane. So whether or not you have a nucleus would distinguish between two different types of cell, prokaryotes or eukaryotes. So a prokaryote, I think you've heard of this term in year 8, I hope you have. But a prokaryote is a single-celled organism that lacks a membrane-bound nucleus. So in other words, it does not have a nucleus. Cells of a prokaryote are called prokaryotic cells. So these are some examples of prokaryotes, and you can see here that the DNA is not inside a circle, it's not inside a structure, but most prokaryotes would have extra structures like flagella and cilia. It's mainly for movement because they don't have like legs or stuff like that. So the way that they move is they just have like a little tail, if you will. And bacteria and archaea are considered prokaryote. So a bacteria is a prokaryotic cell. So it is a cell, it is an organism, it can produce its own proteins, and it can be destroyed using antibacterial. So you want to kill bacteria, you use antibiotics. So antibiotics is for bacteria. A virus is not a bacteria. A virus just has the DNA. It doesn't have the ribosome. It cannot produce its own proteins. I have some examples of bacteria here. Some of them are bad. I think this one's actually good for you. This is a good bacteria, but most of them are pretty bad. And they look kind of gross. But there is a variety in the types of shapes of bacteria that you can have. So things like this stuff, you get it in food poisoning, I think. This is actually multiple bacterial cells combined together. So they join force and that helps them to, I don't know, better attack the host. We're not going to talk about archaea because they're really, really weird. But anyway, eukaryotic cells, I don't know why there's a fungus here. A eukaryote is an organism that has a nucleus. So the difference between prokaryotes and eukaryotes is just whether or not the DNA is inside a nucleus. And the cells of an eukaryote are called eukaryotic cells. There are protists, fungi, plants, and animals. So this is a plant cell, an animal cell, fungus. So I think example fungus would be a yeast, a mushroom. Now, protists, essentially a protist is any eukaryote that doesn't fit the definition of a fungi, an animal, or a plant. But the protists, the different protists don't have a lot in common. So they just eukaryote, but that are not fungi, animal, or plants. But they really, I don't have any other things that I can say about protists that they have in common. Fungi, animal, plants, or animal. Okay, this is an example where protist is Plasmodium. I actually can't remember the name, but this is the malaria parasite. So malaria is actually a eukaryotic cell. Um, it's a unicellular organism, so it's made of one cell and it's a protist. So this is the malaria parasite. So go back to our question, virus, not a living organism. Bacteria is a prokaryote. Malaria is a eukaryote that is unicellular, and it is different from mushroom or animals or plants. In terms of genetic materials, also the types of organelles that they have. There are two more organelles that I want to point out if you compare the difference between plants and animals. So obvious we are here, this is us, we are the animal cells. But plant has this bit, it looks like a pool of water. It is a pool of water, by the way. It also has this, which is green, and it has this. So there are three extra features that you see that is very prominent in plant cells. Fungi kind of look like it has some sort of, it has that as well. But the pool of water is a bit smaller. It doesn't have the green stuff. It looks like it has a thing outside, but the thing outside is actually not very similar to this thing. So we're going to talk about these features. The pool of water, the green stuff, and the outer layer that animal doesn't have. Okay. Oh, before that, here is an example of prokaryotes. Pro is like before, so before nucleus. Karyos, nucleus. Prokaryote, before nucleus, no nucleus. Eukaryote, true nucleus, it has nucleus. Why is this plant so angry? I don't know. So we're going to talk about the green stuff, and the green stuff that is in the plant is called a chloroplast, and this is the organelle that allows the cell to perform photosynthesis. So obviously we as animals cannot perform photosynthesis. That'd be good if we can perform photosynthesis. I no longer have to eat, but we can't. So plants can, so because they have chloroplast. Photosynthesis, for those who have forgotten, is a process that uses sunlight and carbon dioxide to produce sugar and oxygen gas. So chloroplasts are found in plant cells. Some algae, so some protists, also has chloroplasts, and if you ever heard of the term algae bloom, so you have a lot of algae in like the bottom of a lake, and then it's like the lake is like green. It's not good. Anyway, the green pigments that makes the chloroplast green is called chlorophyll. So this is the chloroplast, and the thing inside, they look like stacks of coins. They call chlorophylls. Chlorophyll. Okay. The pool of water is called vacuole, and essentially it is a storage bubble that may store food and water. It's not that animal cells don't have them, but they normally larger in plant cells because plants needs a lot of water for photosynthesis, so it tends to store that water in the vacuole. Because that's the vacuole, it has H2O inside, can also have other nutrients inside, but mainly water. And then the last thing, which is the surrounding structure, that's a cell wall. Some cells have cell walls. The cell walls but the composition of the cell wall can change depending on the type of cell that you have. So if you have a plant cell, you have the cell membrane, but you tend to have, that's a really weird drawing. So this is membrane. But you tend to have an extra layer outside of the membrane called the cell wall. And this is just to provide extra protection, because if you're a plant, you don't have a lot of things that you can do when it comes to protection and against predators or things that want to eat you. Because you're just a plant, you can't run away. Some plants can produce poison and toxin, but cell wall is one of the main forms of protection. And it is a very rigid barrier that provides extra protection for the cell. And fun fact, we actually cannot digest cell walls. And I think that's actually very effective protection. Any organism that eats plants actually cannot digest this. The only organism that can digest cell walls are bacteria. So if you are an animal and you live on a diet of plants like your koala or something, you actually need to have bacteria in your stomach, and the bacteria is going to digest the cell wall for you. We also have some of those bacteria in our large intestine, but there's not a lot of it because we eat meat and other form of proteins. This is why people eat yogurt, so they get more of the bacteria to digest the vegetables that you eat. I have two diagrams here, and this is just to show you where the cell walls are. I don't know whether you did this in year 8, but this is an onion cell, so it's a classic use of microscope. You slice a layer of an onion, and then you dye it, and you will see, and the thing that you can see, this is a cell wall. And then you see, I think, I don't even know what's going on here, but that's the nucleus. And similarly, you can see this one's a little bit better, but this one looks more like a drawing than an actual picture under a microscope. But you can see the cell wall, very, very thick layer, very rigid. And again, you can see the nucleus. I think all of the other organelles are probably too small to be able to be viewed under this microscope. That's it. That's all. This is just a quick recap lesson anyway. You should have learned most of it in year 8, but if you haven't, this is a good chance for you to recap the functions of some of the organelles. So hopefully you can now answer this question, which is what is the difference between coronavirus? Coronavirus is a virus, so it's non-living, doesn't have much when it comes to structure, just has a protein with some DNA in it. So protein, and then you have some DNA inside, or in the case of the coronavirus, actually has RNA. That's a whole other story for another day. And it can't produce its own protein. It has to infect another cell and then force that cell to produce its protein. So it forces the other cell to say, use this instruction, make my protein. Stop making your proteins. A bacteria is a cell, it's a prokaryote, and it's going to have one extra thing that viruses don't have, and that's a ribosome. So it still has the DNA, but it can now translate this DNA into an actual product using a ribosome. So it can make its own protein. A fungus is a type of eukaryote. Malaria is another type of eukaryote. So malaria, a lot of people think malaria is a bacteria, but it's not. It's actually a more sophisticated type of cell. Also eukaryote. And human is another type of eukaryote. But they are slightly different in terms of structure, and the genetic materials is slightly different. So we'll talk about that in a future lesson. But all eukaryotes the main difference between eukaryotes is that the DNA is now inside a nucleus. So you still have your DNA. You can draw the nucleolus if you want. And then you still have the ribosome, but they probably attach to the endoplasmic reticulum. So you have a lot more sophisticated organelles. And then you have the Golgi. And then you have mitochondria. Just label your diagram. I know what you're trying to say. My diagrams just break down this 11 hours of the day. Mitochondria. And last but not least, the lysosome. If you're a plant, you may also have a vacuole that's really big, so it's probably visible, and you have a chloroplast. That's it. That's the difference. Do the worksheet, and I think I have a glossary list because biology in general has a lot of terms, so it's a good idea for you to start building your glossary from day one. So I think you should be able to find the definition to all of these terms throughout this PowerPoint, and maybe just write the terms down and then have the definition next to it, and do the worksheet that I said. Thank you.